Thank you very much. It's really an honor and a pleasure to be here. Before I get started, though, I'm compelled to make a comment about what it means when a scientist wins an award. The work that I'm going to be talking about today, not just the bigger picture of the kinds of studies that we've done, but also the work that I was directly involved in, was not done just by me. It was done in collaboration with my mentors and with other colleagues, and now most recently with students in my own laboratory. And this is now a growing field, and many of the people who've done important work I have not worked with at all, and yet they're making really important contributions. So this really is a collective effort. And my work is only a small piece of that larger story, and it all has been done in collaboration. And I'm going to be, I hope, throughout my talk today, acknowledging the individuals who made contributions where that comes up. But I wanted to, at the outset, just let you know that it really is the effort of many, many people without whom this work wouldn't be possible. So I'm going to, getting ahead of ourselves. I'm going to be talking about this concept of structural plasticity in the adult brain. And what was first mentioned in the kind introduction that Matthew gave of my work was that a lot of my research has been focused on this phenomenon called neurogenesis, and in particular adult neurogenesis, which is the production of new neurons, entirely new neurons, into an adult brain. But that work, which we sort of stumbled upon accidentally in the laboratory, and in the beginning was the result of some studies where we were looking at broader questions of how the brain changes its structure in adulthood. And even today, the studies that are ongoing in my laboratory and many other laboratories have been looking not just at the production of new neurons, but also at other forms of structural change. So I wanted to introduce first this concept of structural change and tell you that adult neurogenesis is just a piece of that story. Another thing that we've been looking at is how pre-existing neurons, or neurons that were produced during development, change their structure. And a lot of this occurs in response to experience as well. So here we have three examples of neurons or neural elements in the brain, in the adult brain, and they're all stained with fluorescent markers, and these are pictures that were taken with a laser scanning microscope. And you can see this is a reconstruction of a very elaborate neuron. The cell body is located in the center, and these extensions are dendrites, or the parts of the neuron that receive inputs from other neurons. And we know that the dendritic tree, the size and the shape of the dendritic tree changes in adulthood. And we know that this is very responsive to experience. So that's part of what I'm going to be telling you about today. Shown in this middle panel is one of these dendritic segments magnified even further. And you can see these protuberances along its extent. And these are something called dendritic spines. And dendritic spines are sites of major connections between neurons. And those also change in their size, shape, and number throughout adulthood. And they're very sensitive not only to experience, but also to hormones. And finally, over on the right-hand side, of course, last but not least, are new neurons, or this process of adult neurogenesis, which most of what I'm going to be telling you about today will focus on. So in this panel, we see these green cells are all neurons that were born in adulthood and incorporated into the existing circuitry. And this was something that was not recognized as a real phenomenon in neuroscience until fairly recently. So what I'd like to do is give you a little bit of historical background to this concept of structural plasticity or structural change in the adult brain, and then bring it to the current day give you some idea about the methods that we use in order to investigate the production of new neurons and other forms of structural change in adulthood, and then focus in on what the function of structural change might be, what all of this turnover of neurons and the changes in the dendritic trees and in the connections between neurons might mean for the function of the brain region in which that occurs. And then finally, I'd like to give you an example of how experience can shape brain structure in adulthood. And I don't have enough time to tell you about all of our studies on this. I'm going to be touching a little bit on our work on stress, but then focusing in on an even more complex experience, which is caregiving or parenting. So to begin with the history of structural plasticity, we need to go back to the turn of the previous century with the work of Santiago Ramon y Cajal, who was the Spanish neuroanatomist, and he was arguably the father of modern neuroscience. And I especially like this picture of Ramon y Cajal, surrounded by some of his children, because he really shows the joys of parenting in this picture. And at the end of my talk, I'll be discussing some evidence we have that shows that fatherhood can alter brain structure. So it's just sort of an interesting prelude to that. But more toward the beginning of Ramon y Cajal's career, he speculated about the possibility that the adult brain would change its structure, and that some of the structural change might be driven by experience and might actually underlie some changes in brain function. And here's an excerpt of some of his writings. In 1894, he wrote, "It can be admitted as very probable that mental exercise leads to a greater development of the dendritic apparatus and of the system of axonal collaterals in the most utilized cerebral regions." Ramon y Cajal spent a lot of time studying the development and regenerative properties of the nervous system of several different species of vertebrates. And several years later, he essentially reversed his position and wrote, "Once the development was ended, the founts of growth and regeneration of the axons and dendrites dried up irrevocably." So I don't want to leave you with the impression that Ramon y Cajal was wrong about everything, because he really was the founder of the field, and many of the discoveries he made have absolutely stood the test of time. But in this particular, on this particular topic, he ended his career on a negative note, and largely due to the fact that the techniques were not available in order to actually see brain structure changing in adulthood. He was inaccurate in his conclusion. And at this point, there really wasn't much work that was done on this topic at all. And it wasn't until many decades later, in the 1960s, when Mark Rosenzweig and his colleagues at UC Berkeley did a whole series of studies where they raised rodents and raised rodents during development and also housed rodents in adulthood in the so-called enriched environment conditions. These conditions were just larger cages where they had access to running wheels and toys to interact with. And when they compared the brains of these animals to the brains of animals that lived in standard laboratory settings, they found that their brains were larger, the neurons were larger, and they had greater dendritic trees and had more dendritic spines. So this was in the 1960s. There was a whole series of studies that were done on this topic. Around the same time, Joseph Altman and his colleagues at MIT reported evidence for adult neurogenesis in the hippocampus, olfactory bulb, and neocortex of rats, guinea pigs, and cats. And this was a series of about 6 studies, 6 papers published in high-profile journals, but they had little, if any, impact on the field. Fast forward a bit to the late 1970s and early 1980s, Michael Kaplan and his colleagues at Boston University revisited this question and provided further evidence of adult neurogenesis in these 3 brain regions, in rats this time, and this evidence was actually quite strong. He used an electron microscope to identify the new cells as neurons, and there was about 6 papers published in very worthy journals, in high-profile journals, and it really had little impact on the field, perhaps because the time was not ripe for people to actually accept this as a real phenomenon. In the 1980s, Fernando Nottebohm and his colleagues at the Rockefeller University reported evidence of neurogenesis in the adult canary and zebrafinch brain, and this work was actually very well accepted, and it was very beautifully done, and they not only showed that the new cells looked like neurons, but that they actually were functioning like neurons. It was a very strong set of data, and it was accepted perhaps because people were willing to consider the possibility that a bird, which was very different from a human, might have a lot of structural turnover because it needed to keep its brain light in order to support flight. And there was a lot that was written about this. That adult neurogenesis in the case of the bird was a possibility because it was a specialization of some of the unique features of being a bird. And around the same time, a very influential paper was published reporting no evidence for adult neurogenesis in any brain region in macaque monkeys. And at this point, this was published in 1985, at this point the field really stopped work on this topic. And there was really nothing that was done for quite some time. And it wasn't until the 1990s when I joined Bruce McEwen's laboratory at the Rockefeller University that we started to look into this question quite by accident. And I'll just give you the short story of how this happened because I think it's easy in retrospect to say when you're involved in something that ends up being influential that it was intentional. And at some point it did become intentional, but I think it informs us a little bit about the scientific process to know that some of this really was sort of stumbled upon by accident. We were studying the influence of hormones on brain structure, not on neurogenesis, but looking on the survival of new cells and how they depended upon certain kinds of hormones. And we found that when we performed a certain hormonal manipulation, which was taking out the adrenal glands, that in a certain part of the brain there was a massive death of neurons. We could see this with a microscope. And yet when we counted the numbers of neurons that existed, we found that despite the fact that thousands and thousands of cells were dying, there was no net decrease in the numbers of neurons. And this, we were trying very hard. This was very confusing. We were trying to understand it. And we eventually dug up the old papers of Joseph Altman from the 1960s. Remember, this was before the internet was available and before scientific papers were available on Medline and other search engines. And we were able to find these papers and do studies looking at hormone regulation of neurogenesis. And so it turns out that the reason we could not find this decrease in the number of neurons, despite the fact that cells were dying, was because there was compensatory neurogenesis in this region. And that started us off into studying this different form of structural plasticity, which, because these earlier studies really had not been influential, the work had been suppressed up until this point. And then we started to look using newer methodologies. And around that time, around the mid to late 1990s, a lot of other laboratories started to do this as well. And this new methodology really enabled the field to come to the realization that these were neurons that were being produced, and that they were being produced at a relatively high rate. And that suggested that they might have some functional consequence. So I want to take a moment and just describe this methodological development because it really, I think, did play quite a big role in the field coming to accept adult neurogenesis as a real phenomenon. The older method that was used by Altman and Kaplan and also by some of our early studies was this tritiated thymidine method, which is a radioactive method whereby you provide a radioactive nucleotide to the brain, and it's picked up by cells that are synthesizing DNA in preparation for division. And then you can determine which of those cells picked up this radioisotope afterward, and you can do this using a method that's very similar to traditional photographic development. And you can see this cell that has the black silver grains over its nucleus is a cell that picked up this nucleotide in preparation for division, hence it was born in adulthood. And this this new cell is very similar to the cells that were reported in those earlier studies. One of the weaknesses of this methodology is that the radioisotope that's used, tritium, is very, very weak, and it can only expose labeled neurons that are on the top 1 to 3 microns of a tissue section. So unless your sections are very, very thin, you're going to underestimate the numbers of neurons or new cells that are produced, and that's exactly what was happening with the earlier studies. A bigger criticism that was levied against this method is the fact that you cannot determine with certainty whether the cell that you're looking at that's labeled with these black dots is actually a neuron. And it's always possible, even if it looks like a neuron, that it could be a non-neuron cell that picked up the tritiated thymidine because it was made in adulthood and it's lying on top of a neuron. So that when you look down in one plane through the microscope at this labeled cell, it looks like a labeled neuron, but it really isn't. It's 2 cells. And this was a major problem in the field, and it was not solved until the advent of this new technology called BRDU, or bromodioxyuridine labeling. It works a very similar way to tritiated thymidine, but you don't need a radioisotope in order to look at the cells that are labeled. You can provide this BRDU molecule to the brain. It's picked up by cells that are about to divide, that are synthesizing their DNA, and then you can visualize it using antibody methods, something called immunohistochemistry. And this brown cell is an example of a new cell in the same brain region as that is shown over on the left-hand side. And it's labeled with BRDU, indicating that it was made in adulthood, and given where it's located, and its shape, it's most probably a neuron. However, we don't know this for sure. In order to do this, to ascertain this, we need to use immunofluorescent methods. So we need to label these cells with a fluorescent marker, and then label the cells, label the tissue with markers that are only expressed by neurons, and then use a laser microscope in order to take very, very thin sections, optical sections, through the cell, reconstruct it, and rotate it in 3 dimensions in order to make sure that it is not a non-neuronal cell that was new lying on top of an old neuron. And that's exactly what we can do now. So I want to show you this. One particular example. In this case, we know that the cell is new because it's labeled with this red stain, which stains the nucleus of cells that have made their DNA around the time when the BRDU is available. The green marker is a neuronal marker that's called NeuN, which is just made by cells that are neurons. And what we can see here when I rotate this cell is that there's no point at which the red is separate from the green. You can see the red and the green coexist throughout the extent of the labeled cell. And it was evidence like this that convinced the real skeptics in the field that there was, in fact, adult neurogenesis in certain brain regions. And now we know, particularly for one brain region that I'm going to focus mostly on today, which is the hippocampus, that there's very good evidence that these new cells not only attain neuronal characteristics, but actually become integrated into the existing circuitry. We know they have synapses, or connections with other neurons on their cell bodies and dendrites. We know they make dendrites, and they also extend axons, which is the way that it's the part of the neuron that enables the neuron to communicate with other cells, to send information away. They generate action potentials, which is an electrophysiological signature of a neuron. And they also exhibit enhanced synaptic plasticity, which means that their connections between neurons can be strengthened in such a way that it's actually greater than what we see with mature neurons. So it looks like these new cells actually in some ways are stronger than cells that were produced during development and are fully mature. And of course, we know that they express these neuron-specific proteins. And you can see some other examples over to the right. In this case, the new cells are red, shown it's the same microscopic field. And down below, we have the neuronal marker shown in green. And you can see that there's coexistence of the neuronal marker with the BRDU label, demonstrating again that many of the cells that are produced in adulthood differentiate into neurons. So on the basis of the collective work of many, many labs, we have some indication of how this process occurs. We know that there are cells with stem cell-like properties that are located in the hippocampus itself, and we know that those cells divide or proliferate. They migrate relatively short distances where they become incorporated into the layer that consists of all of the mature neurons. And once they migrate into the layer, they extend dendrites as well as axons into target regions, and they also begin to express neuron-specific proteins. So one question that's important to ask is how many new neurons are produced in adulthood? And this is an important thing to ask because if we're going to speculate that these new neurons might participate in some functions of the brain region to which they're incorporated, there have to be a substantial number of them. We don't have really good quantitative data on all of the species that have been investigated with regard to adult neurogenesis, and most of the data that we have are taken from rodents, but there's good reason to believe that there are similarities between rodent and primate models, which I'll tell you about in a moment. And this work by Heather Cameron and Ron Mackay demonstrated that there are over 9,000 new neurons produced every day in one brain region in the young adult rat. And that extrapolates to about 250,000 new neurons a month. And if you consider the fact that this particular brain region, the hippocampus, and the subregion into which the neurons are incorporated, has between 1.5 and 2 million neurons contained within it, that's a substantial proportion of the total population of neurons. And if you also bring into that story the fact that the new neurons have very robust electrophysiological responses and actually are stronger in some ways than the mature neurons, it's very likely that they're having an influence over the existing circuitry. So the next question that comes up that's been another problem that the field faced is whether or not this is a general characteristic of mammals. I told you about the earlier work by Fernando Nottebohm, which was relatively readily accepted, and the work by Joseph Altman and Michael Kaplan really focused mostly on rodents. And most of the work that was done in the 1990s that I referred to, work that I did with Bruce McEwen and also work that Fred Gage at the Salk Institute and other groups have done, focused again on rodents, rats, and mice. And the question always remained of whether or not this was just a feature of less complex animals. And if so, it wouldn't necessarily be so important to study unless we were interested just in understanding the brains of less complex animals, because if you could not demonstrate it in more complex animals or animals that were more closely related to humans, then it really was unlikely to be relevant to the human condition. So we set out to do a series of studies to investigate this question, and in collaboration with Eberhard Fuchs at the German Primate Center and Bruce McEwen, we looked at tree shrews, which are insectivores, and found that this creature has a substantial amount of adult neurogenesis in the hippocampus, very similar to what we see with rats and mice. Then with Eberhard and Bruce, we looked at marmosets as well. Marmosets are New World monkeys, and found a substantial amount of adult neurogenesis in the dentate gyrus. And finally, we looked at macaques, which are Old World monkeys. This was done in collaboration with Charlie Gross at Princeton, and we found, again, a substantial amount of adult neurogenesis in the hippocampus of these animals. And this collectively suggested that this was a general phenomenon, a general feature of mammalian brains. Around the same time, Peter Erickson and Fred Gage did a groundbreaking study looking for adult neurogenesis in the human brain. And this is a very difficult thing to do because we don't routinely inject molecules like BRDU or tritidothymidine into humans. Peter Erickson was in Sweden at the time, and in some Swedish hospitals, they were treating individuals with BRDU in order to assess the proliferation index in their tumors, and these people had throat cancer. And then when the individuals died, their brains were donated to this study, and they were examined to see if there were, in fact, if there was evidence of adult neurogenesis. And what they found in a relatively limited number of patients, because that was all they had access to, that in every case there was evidence of adult neurogenesis in the hippocampus. And this was even in very old individuals, as well as and all of them were very sick and dying of cancer. And we know that aging is associated with the decline in neurogenesis, and we know, as I'll tell you in a moment, that negative stressors can decrease neurogenesis. So this really suggests that this phenomenon of adult neurogenesis not only occurs in humans, but probably in young, healthy adults, it's a relatively robust phenomenon. Now, that was only one study, and it was just done, and it's not likely that that method will be useful in investigating this phenomenon in humans in the future for reasons that I told you before. And our neuroimaging capabilities are not yet of high enough resolution in order to actually look at new neuron formation in the human brain, but I'm definitely aware of the fact that there are studies that are ongoing to develop new technology in order to do this. But at this point, we just have that one study, and we really don't know whether there is adult neurogenesis in all humans. So what are the functions of the hippocampus? And this is an important question to ask when we're trying to understand whether structural plasticity actually plays an important role in function of the brain and also in behavior. And since most of the adult neurogenesis that the work that I'm going to be telling you about focuses on this brain region, it's very important to talk about and think about the possible functions in light of what we know about this area. And it's an area that's been studied intensively for over 50 years in neuroscience. This is just a drawing of the hippocampus showing its involuted structure. And we know that it's very important for certain types of learning and memory. We know that it's important for anxiety regulation. We know that it plays an important role in stress, and in particular feedback of the stress response, so shutting off the stress response once it occurs. And we also know that this brain region has been implicated in a number of psychiatric and neurological conditions, including post-traumatic stress disorder, depression, and dementia. In all three of these conditions, the hippocampus has been shown to be smaller in numerous studies. So we can ask the question, because we know that the hippocampus is a site of a lot of structural change, not only adult neurogenesis, but dendritic remodeling and changes in dendritic spines, of what might this structural change be doing. And many groups have been looking into this, and there's some evidence, although the picture is really just emerging now, and we don't have a clear and precise story, but there's good evidence supporting the general claim that structural plasticity is important for certain types of learning and also for anxiety regulation. And we know this through at least two different lines of evidence. The first is that learning and stress are known to change structural plasticity. And this suggests to us, and some of the earliest work done on this topic looked at this possibility, and it suggests to us that if these experiences can change the structure of the hippocampus, then it's possible that that structural change is underlying some of the functions of the brain region that are associated with those particular experiences. And from work that Ben Lunar, a postdoc in my lab, and Chris Morescu, a grad student in my lab, did, we know that learning, both learning and stress, are associated with changes in the numbers of not only of dendritic spines, but the numbers of new neurons in the hippocampus. And we know from several different studies done by my lab in collaboration with Tracy Shores at Rutgers and by other laboratories, that if you deplete the brain of new neurons, so if you stop adult neurogenesis, you can alter learning in some situations, and you can also alter anxiety regulation. Now, these studies, to be fair, are not without their criticisms, and the literature is rather mixed on this subject at this, to this date. But there are many, many studies, and I didn't have enough space to cite them all here, that support this general idea that the brain shows altered learning and memory capabilities if you stop adult neurogenesis. And there are fewer but a growing number of studies that suggest that there's a link between new neurons and anxiety regulation as well. So there's some evidence, and I suspect that in the coming years there will be even better tools developed in order to investigate this, showing a link between structural change and hippocampal function. So I want to turn for the rest of my talk to discuss this, some examples of how experience can shape the adult brain. We and others have done a whole series of studies looking at the effects of aversive stress on the structure not only of the hippocampus but of other brain regions. And we found that both stress and elevated stress hormones causes a decrease in adult neurogenesis, a decrease in dendritic complexity, a decrease in dendritic spines, and a decrease in the numbers of connections between neurons or synapses. And this is perhaps not surprising because stress hormones are known to be catabolic in nature. They are known to inhibit growth and to enable energy mobilization so that the individual can cope with a stressful event. So showing that there is a growth suppression is not surprising, but there are some interesting counterexamples that we've stumbled upon in our investigations where glucocorticoid or stress hormone levels are elevated after certain experiences, and the brain is growing nonetheless. So this is not just a universal phenomenon. It seems to be something that happens when animals are exposed to aversive experiences that are aversive across the board, an experience that an animal will escape from if it's given an opportunity to, and experiences that animals will never choose to engage in. If an experience is more complicated than that, and it still increases glucocorticoid levels, but it has a rewarding component, it is often associated with neuronal growth. And one of the most complicated experiences that we've looked at in the laboratory, and I want to spend a moment to tell you about it, is that of parenting. In order to do this, we've looked at 3 different creatures that have different caregiving strategies. We've looked at the standard laboratory rat, which is an exclusively maternal animal, meaning the mother is the only caregiver of the developing rat pups. And in fact, if male rats get near the maternal nest, if they get access to the pups, more often than not, they'll commit infanticide. They really don't play any substantial role in rearing the young. There are some rodent species, it's rare, but it does exist, where the father does participate in caring for the babies. And shown here on the right are an example of a California mouse, which is a field mouse family. This is the father and the mother and the pup down here. And these animals are biparental. The father takes care of the babies as much as the mother does. The only thing that he does not do is nurse the babies. So he will huddle over the babies, he will lick them and groom them, he'll retrieve them, and he engages in nest building. And you'll also notice that these animals have many fewer pups than rats do. Rats can have up to 12 to 14 pups, and these biparental animals have 1 or 2. And they put an awful lot of parental investment into the small number of offspring. Very unusual, but certainly a little bit closer to what we would say is the way that most human communities raise their young. And then finally we looked at marmosets, and marmosets are cooperative breeders. And the father participates in taking care of the babies. This is a marmoset father with some young on his back. And in fact, during the daylight hours, the marmoset father will carry the babies 70% of the time. So he's actually doing more of the caregiving than the mother. It's not true at night, because that's when the babies primarily nurse. But he does participate quite a bit. And interestingly, it's not just the marmoset father and mother that take care of the babies. It's whatever other larger marmosets are present with a family group. So it can be related and unrelated marmosets, it can be juveniles, it can be full-fledged adults. If they're living in the same group, they'll participate in caring for the babies, in carrying them and in feeding them when they get closer to weaning. So it really is a cooperative breeding effort, and it's very similar to what goes on in many human societies. So we were interested in looking at the influence of parenting on brain structure, and one of the reasons we were interested in doing this was not just because of our long-standing interest in how hormones shape the brain, but also because parenting is a very complex experience, and it has both stressful component and enriching component to it. And we had, some of this existed in the literature, and we also had observed this in some of our behavioral studies, that there's a very strong hedonic component to parenting. So, for example, a mother rat that's been treated with cocaine for a relatively long period of time will actually choose access to her pups over access to cocaine, because there's a very strong rewarding component to parenting. And you can observe that with these biparental creatures as well, that often there's fighting in the family groups over who will actually have access to the babies. So the fact that this is stressful but also rewarding was very interesting to us. And we embarked on a whole series of studies that are ongoing now in order to look at what this was actually doing to the brains of these animals. And I'm just going to give you a little bit of these data to give you an idea of what we're thinking about now. This is the first study that was done by Ben Lunar in my lab, and she looked at mother rats, and she found that they produce many fewer neurons during the postpartum period. So this is just a graph showing the numbers of new neurons, and the white bar shows what the animals are like before they have their babies, and then the gray bars show what the animals are like just after they have the baby and as the period of lactation continues, and then around this time the animals are weaned, and they start to look very similar to the way that they looked beforehand in terms of the numbers of new neurons that are in the hippocampus. And these are just photomicrographs showing new cells in the hippocampus. And we also know that during this period, animals show changes in behavior and in certain functions that can be attributed to the hippocampus. So we know that postpartum females have changes in anxiety levels, and they also are impaired on certain learning tasks, but better on others. And we don't yet know whether this structural change, what functional change it's responsible for, but we're investigating that now in the laboratory. Ben went on to determine what was the mechanism that underlies this effect, and she found that stress hormones were driving the effect. Postpartum females have elevated stress hormones, and stress hormones are very important for normal lactation. And she found that if stress hormones were normalized in these animals, there was not a dip in adult neurogenesis. So this seems to be driven by an increase in stress hormones, which is linked to lactation in these animals. Given that information, we hypothesized that this particular structural change would not be seen in fathers, because although in those creatures I described for you, the fathers are participating in the care of the young, they're certainly not nursing the babies. And we were very surprised when we looked into this to find that fathers also produce fewer new neurons in the hippocampus. And we believe this is also linked to elevated glucocorticoids, but obviously not tied to lactation. But the story is not only negative, there's a positive side to it as well. Although the fathers and mothers are making fewer new neurons, a greater proportion of the neurons that they are making seem to be activated in these animals. So it looks like more of the cells are being incorporated into the existing circuitry and are being used by the animal. And this is just a graph showing the proportion of new cells that express a molecule that we know is an indirect marker of neuronal activation. It's called CFOS. It's an immediate early gene. And you can see in the fathers there's an increase in those numbers. When we look at other types of structural plasticity as well, we see evidence for neuronal growth in parents. We see this in both mothers and in fathers, but I'm only going to show you one experiment that we did. We looked at marmoset fathers, and here's an example of a neuron labeled with a lipophilic dye called dye I. and it's located in the prefrontal cortex. And what we found was that there's an increase in the numbers of dendritic spines, or those sites of connections among neurons, on the prefrontal cortex neurons of animals that are engaging in caregiving behavior. And this is work that Zhenya Kozarovitsky, a graduate student in my lab at Princeton, did. So the fathers had more dendritic spines than the naive animals, or than the animals that had never had babies. And there was no difference between first-time fathers and experienced fathers, so it looks like it's a one-time change that occurs. Zhenya also found that this occurred in not only mothers, but also in juvenile caregivers, so in the older siblings of the babies, because they're also participating in caring for the babies. And she found as well that housing animals in an enriched environment caused the same kind of neuronal growth in this and other brain regions. And that really suggests that parenting has this sort of positive enriching result on certain brain regions, in addition to the fact that there's some stress-related phenomena that are going on that are potentially inhibiting growth in other areas. We don't know for sure what the functional changes are that are associated with this, but tasks that are associated with the prefrontal cortex have been shown to be altered. Animals seem to be better at them after they engage in caregiving behavior. That's work that Ben Lunar did in looking at mother rats that also show this growth in the prefrontal cortex. So it looks like there's at least a correlation between these structural changes and the functional change that we see. So just to summarize here, we know that the hippocampus, as well as other brain regions, remain structurally plastic throughout life. Experience can alter adult neurogenesis and dendritic architecture, and this sometimes happens in very complex ways. And finally, structural plasticity may be important for learning and anxiety regulation. And I want to end by just thanking the most recent and current members of my laboratory, without whom this work would not have been possible. I mentioned most of these individuals in my talk, and I thank you also for your attention. So I'm going to stop there.